So the next few moments, I just want to set a little bit of a context of our thinking around why we believe the world should change from disease care to health care. And I think it always, whenever we have these conversations, so it's nothing new, it always starts with patients. And so if you bear with me, I'd like to share with you the story of Kaisi Terry. So Kaisi was born in October 2009. You can just look at her picture. You can see that she was a young girl, young lady full of energy. She loved gymnastics. She loved cheerleading. If you talk to her parents, they would always say she was the center of attention wherever she was at. Not only was she the center of attention, full of energy, when we visited with them, they said they never remembered her being sick a day in her life. And so her life was such that she had just a little over 5.5 on a January morning. It was a Sunday morning. As Kaisi did with her family, she went to church as a normal morning would. She came home after church and she really wasn't feeling the best, had a little bit of a headache, and so her parents said, "Why don't you take a nap?" Which she did. She woke up, was feeling good enough at the end of the nap to play games with her brothers, which she always enjoyed doing. And then went to bed. The next morning she woke up, wasn't feeling the best, and her parents said, well, she must have the flu. And so they said, why don't you stay home from school on Monday, which she did. And on Tuesday, when her brothers came down with what they thought was the flu as well, they said, yes, our confirmation she does have the flu was right. And so they all stayed home from school. But unfortunately, on Thursday, when her brothers felt better and went back to school, Kaisi didn't. And on Friday, after not eating almost for a week, or barely eating for a week, they took her into the emergency room where they quickly identified her with diabetic ketoacidosis, a blood glucose level of 1,068. They put her on a helicopter, rushed her to Salt Lake City. She was living in a small town in Utah. Unfortunately, on the way, she suffered seizures, and Kaisi never saw her 6th birthday. Now, all of us know that Kaisi wasn't feeling great on Saturday, perfectly healthy, and overnight came up with this disease of DK, which obviously then is type 1 diabetes. A number of us, as Paul said in his opening thoughts, right, we have diseases that are incubating, but most of the time we don't do anything about it until the clinical manifestations appear. And I think all of us are challenged to say, why do we wait? In fact, we believe that 100 years from now, people will look back on us and say, can you imagine there was this time in life, a time in healthcare, where people waited till they had a disease before they did something about it? Almost as if that was just like, it would be like unheard of thinking. And that's what we're trying to solve in what we call the World Without Disease Accelerator. Now, why is that a problem? Well, the problem is today, as you know, the good news is most of us are living longer. That's positive. The downside is as we live longer, many of us gain, I call little baggages along the way, if we could call them that, little comorbidities. And as we get older, we get more and more of those, right? And so as you get to be in your 70s, the majority of people have 2, 3+ comorbid conditions. And so if we basically say that we're going to live longer, but we don't change the trajectory of health, the promise is that we're going to live longer, unhealthier lives, which isn't attractive, I don't think, for any of us. But the question is, as you think about with Kaisi, as you think about maybe some of our parents or some of the friends that we know that all of a sudden present with a disease, all of us are, if we could say it this way, incubating disease in some way, shape, or form, but we're not sure what it is because on the outside, how we normally look at people or how we feel ourselves, we feel healthy, but what would happen if we could truly identify these diseases that each of us are incubating and could address it before they manifest with clinical symptoms? And that's what spawned about 5 years ago at Johnson & Johnson and the Janssen group of pharmaceutical companies, this concept of we envision a world where disease becomes a historical artifact. It's a phenomenal vision. It has a lot of other consequences, which I'm more than glad to talk to you about the break. But we feel like to really put out a bold statement, to really change the course of disease, we need to think boldly. And so that's our vision. Now, how are we going to achieve that? Well, what you heard from Paul is, let's start at the beginning. And the idea is if people are healthy, let's do all that we can to keep them healthy. And that requires what? That requires different skill sets than what we maybe would have down where we're treating disease. It requires our understanding of nutrition. It requires our understanding of behavioral science. It requires our understanding of early detection, of the immune system, of the endocrine system, of the neurological system, and doing all that we can early in life to keep people healthy. But also, if for some reason someone veers off course a little bit and starts to incubate a disease, and we can do that by probing the different systems we now know, and we are now gaining more and more understanding on a day-to-day basis, and we can understand changes, which we're going to hear later about, in the immune system that could be picked up very, very, very early, and we can identify those people headed toward disease. Let's intercept the disease. So going back to the story of Casey, we know today that if someone has multiple autoantibodies to a wide range of insulin or other beta cell proteins, they will have type 1 diabetes with a 100% chance in their lifetime. The question is it 2 years, 4 years, 5 years, or 10 years? But they will have it. So the question is, even though they're not insulin dependent at that point, but they have the presence of 2 autoantibodies, should we wait for them to have a clinical manifestation to treat the disease? Or should we intercept the disease as we can see it before there is an onset of this clinical morbidities? And then, let's just say we can't keep people all the way healthy, or we miss it for some chance, and we can't intercept the disease, then our goal truly is to cure the disease versus treat the symptoms. A different paradigm requires different technologies, both in terms of measurement, monitoring, and intervention. And at the same time, as we'll talk about later, will require potentially different business models and different regulatory models for us to be truly successful. But if we focus on truly the transformational medical innovation where we can make a significant difference in people's lives, so what are we doing and what are we going to talk to the panelists about? Is at Janssen, we're focusing on diseases where we can make a difference, where we understand the core disease pathology so that we can probe the system to understand where you're at in the course of disease and truly make a difference there. And we understand that some disease areas and some disease specific diseases we're further along than we are in others. And so let's start where we can be pulled by the science. Second is we don't believe we can do this by ourselves. It requires what we're calling a coalition of the willing. It requires other pieces of the ecosystem to work together, whether it's the diagnostic piece or the digital therapeutic piece or the nutritional piece, that we can provide holistic solutions to individuals so they can incorporate and integrate those into their lives so they can truly prevent, intercept, and stay healthy, keep the healthy people healthy. And then the third is thinking about business model innovation. Right? We're not asking people to no longer pay for a product or normal fee for service, but how would we think about sharing risk, or how would we look at new models that will reward healthy performance or overall health of an individual, no matter what that looks like? And so just as we see in the other industries, whether it's the entertainment industry or the music industry, as new transformational innovation came there, it also came along with new business model innovation. And part of what we're working on, and we'd love to, again, an invitation out to this audience, and we'll talk to our speakers here, our panelists in a second, is to help us to think through what are some of these business models that can help spur. Because what we know is when we have solutions that work, whether it's the case of the statins or the osteoporotic drugs, or think of work that's done in Hep C for liver cancer, the system rewards it. So you need to start with innovation, but at times you may need new business models. So we believe the patients are waiting, and we're here to help them live into what we believe is a world without disease.
We've looked at this paradigm from a very simple principle that the system follows, and which is self versus non-self discrimination. That's the essential decision the immune system has to make at the highest level when it encounters a signal. So autoimmune diseases is essentially inappropriate recognition of self as non-self. Susceptibility to cancers or infections is inadequate recognition of something that's foreign and you should react to. That's a fundamental principle. And the reason I focus the entire R&D organization on that is if we understand that, it gets us to the why, not the what. We're not interested in singular molecules and singular cytokines. They've been tremendously successful. We want to make the next leap forward. And the question we ask ourselves is if we can get to the basis of these questions, does this put us in a different space? And the answer is yes, it does. It becomes transformational for the patient and the disease. And here are some examples exactly to what you were talking about. So if you look at the T cell repertoire, this is the diversity of T cells that all of us harbor in our bodies, anywhere between 10 million to 25 million different kinds of receptor specificities that are essentially there to recognize a diverse array of antigens. You don't know what tomorrow's Ebola is going to look like, or 10 years West Nile is going to look like, so you want to be prepared. And what you see on the left is a panel of an autoreactive T cell repertoire on the y-axis as a function of time with age. We all harbor these specificities. In most of us, they're just never triggered. You have a key decision point that's marked by number 1, where you've got an environmental insult. Something happens, an infection, stress, something you encounter that activates that particular T cell repertoire against your self-tissue, so start recognizing it as foreign. That earliest inflection point, I believe, if we could recognize that and track it, which is doable with the immune repertoire, puts you in a whole different space than the chronic stages of disease. There is a stage between number 1 and number 2 where that repertoire is expanding. It's on a slow burn. It's literally a clonal expansion, 1 to 2, 2 to 4, so on and so forth. And at some critical point in that space, if you could intervene, then you can see by that dotted green line, you bring it back to baseline. You've turned the course of disease. Where you start to show symptoms is after the stage of number 2, where you have frank tissue damage, signatures of inflammation, and that's what we're trying to address at this point. We believe with the platform we have, which is a novel biologics platform, it gets to this point. Similarly, if you look at the other spectrum, now we're talking about susceptibility to cancers, the decision is exactly the same, except now it's in reverse and benefits the host, which is you want an anti-tumor T cell repertoire to be triggered when you've got a malignancy that's detected in the earlier stages that provides sterilizing immunity. And we know this can happen in the small number of patients that respond positively to checkpoint therapies. In some cases, you've got lasting complete responses. People don't like to use the word cure in cancer immunotherapy, but when you're off drug and you're going past decades, and I used to be at BMS in the early days of ipilimumab and never very fortunate to have had that experience, you're seeing this. So your immune system has the potential, and you can harness it to the extremes. And in this case, as you ramp up the repertoire to the particular determinants of the tumor antigens, you should see control of tumor growth. I would argue in most cases, as we're looking at therapies for cancer immunotherapy, we still don't have a very good understanding whether the individual primed this in the first place or not. So going in with molecules that unleash the immune system without fully understanding what the breadth is becomes very important. Where I think the challenge is in understanding what are the sensitive diagnostics and where do you sample them. The best way to do this is, of course, in blood. In cases of autoimmune diseases, particularly the poignant case of type 1 diabetes that you mentioned, these can be sampled in blood. There may be certain instances for other diseases we may need other sites to sample from. That may be disease-specific, oriented. I think correlating the specificities back to the therapeutic becomes an important challenge as to how quickly do you translate that information and generate the therapeutic. There's something we're faced with ourselves. If you gave us 10 different tumor targets, can you prosecute all in parallel, or do you? So there's a whole innovation, I think, around platform and protein engineering that has to accompany the biology and the thinking there. But I do think in the next I'd say about five years or so, we're going to see a lot of movement in this space. And I think the trend is going to be likely driven by cancer immunotherapy simply because of the ramped up need. But I would say the vast, enormous opportunity in autoimmune disease and in inflammation is still to be fully recognized. And I come back to the concepts around neurodegeneration, neuroinflammation, metabolic inflammation. These are been diseases that have conventionally not been defined in a particular immune context, but more and more are coming forth. And we're appreciating these mechanisms underlying this inflammation. If you actually sort of think about it, metabolic type 2 diabetes even, if that comes back to this concept of recognizing something that's aberrant, there's no reason to believe, there's no reason to think that one could not apply these in that sort of setting.
In the last 5 years, we've seen a dramatic evolution in imaging of the chest as a better way of finding lung cancer earlier. So for those of you who don't know, CT scans of the chest are now standard of care for screening high-risk individuals for lung cancer. That's really just in the last 5, 6, 7 years. So that's very exciting. The challenge remains that on those CAT scans of the chest, we find a lot of non-specific findings, so-called nodules, most of which are benign. A small number of them actually represent early-stage lung cancer. And it's very difficult for us as clinicians to distinguish a benign from a malignant small pulmonary nodule on a CAT scan of the chest. So what we're beginning to see, and I think we'll see more of in the next, let's call it 2 to 3 years, Ben, is evolution in machine learning and AI algorithms for predicting whether those nodules on a CAT scan are likely benign or likely malignant. And molecular biomarkers that follow, non-invasive tests in the blood that can help, or in the airway, that can help decide whether that nodule needs to undergo a biopsy and resection or is one that you can follow non-invasively with serial CTs of the chest. And I think we're also going to see enormous advances in robotics and ability for us to get out into the lung non-invasively or less invasively and sample that nodule and ultimately treat it less invasively than we do today. So I think all of this is coming in the next 2 to 3 years. And if we detect lung cancer at stage 1 or 2, and I would argue 5 years from now, Ben, we could be detecting 70% of patients at stage 1 and 2 instead of 70% today at 3 and 4. And if we do that, the 5-year survival for this disease can dramatically change. If we detect it at stage 1 today, the 5-year survival is above 60% as compared to 18% overall. So that's the diagnosing early. I think the longer term and more challenging, but again, the one I'm equally excited about is finding it before it starts and intervening before you have invasive disease. And the analog here, and you alluded to this in your introduction, Ben, is cardiovascular disease, right? And I sit across from a cardiologist over at Boston Medical Center, and they're doing it right? They don't wait for someone to have a massive heart attack or massive stroke to begin to treat the patient. They treat the patient years or decades before that heart attack happens. What enabled them to do that? Well, 20, 30 years ago, they got to root cause of disease. They figured out that rupture of a cholesterol plaque is the inciting event for a heart attack. Once they had that fundamental insight, they could then develop less invasive biomarkers of the process in blood. Cholesterol is a great example, to measure the process, and then develop drugs that could inhibit the synthesis of cholesterol, statins. And now we prevent and intercept that disease. So I would argue that's exactly what we are going to be able to do for lung cancer in the next 5 to 10 years. We're going to get to root cause. We're going to do studies that we could never even imagine 10 years ago, where we collect pre-cancer cells from the airway and lung of patients who are incubating lung cancer. They're not there yet, but they're on their way, and bring the latest and greatest molecular technologies, immunological studies like Anish mentioned, to better understand what are the earliest cellular and molecular events that take a normal lung cell, a normal epithelial cell, and convert it into a precancer and ultimately into an invasive cancer. And once we have that fundamental root cause, we can then develop the right drugs, the right interventions to block that process. And equally important to that root cause understanding are biomarkers that are non-invasive that we could measure to follow that process. So putting together large cohorts of high-risk individuals for lung cancer, where we follow them over time, measure those biomarkers, and validate that they're predictive of future events is going to be equally critical. We need that Framingham-like study for lung cancer to get to a cholesterol-like test. But I do believe, back to one of your earlier slides, Ben, that 20 years from now, we're going to look back and say, I can't believe we waited for someone to have an invasive lung cancer before we started to treat them. Even as big as Johnson & Johnson is, it's necessary but not sufficient to really change the bar here and transform that patient journey. We are partnering with academics all over the world to put together those cohorts of individuals followed over time where we can develop that cholesterol-like test. again, we're partnering with academics all over the world to find cohorts, rare cohorts of patients incubating lung cancer with pre-malignant cells that we can obtain to study with the latest and greatest technologies we have today. We're partnering with biotechs. Anish mentioned those 55 people groups that are really innovative, that can move quickly, that can take risk, and quickly bring new solutions to market that are the next statin for lung cancer. So I think it's going to take, as you said, a coalition of the willing, and working again with third-party healthcare payers to figure out what are the right business models so that whatever we do bring to market has a chance to move forward in the real world. So it really is a coalition of the willing.
A couple years ago, we started looking at how to do things a little better in diabetes. And this was actually an idea that we're working on with Janssen. We had this hypothesis that you could look at the underlying insulin resistance and beta cell function by simply analyzing CGM curves, continuous glucose curves. Within those curves was all sorts of first derivatives and second derivatives and things that are indicating how the body is working. It was a hypothesis. We didn't know if we can do it. We sponsored some trials in conjunction with Janssen to develop the data set to do this. And it looks now like you can probably do it, that you can use these curves to, we affectionately called it the clamp without a clamp when we began this, because we wanted to get things that traditionally you can only get with a euglycemic clamp. As we were doing that, we started learning that those signals are amazingly motivating to type 2 diabetics, and they cause significant behavior change. So we did a little bit of an experiment about when people should walk. So for years, the advice on walking has been, I think, equivocal in almost every sense. Should you walk before a meal? Should you walk after a meal? How much should you walk to get an effect? I mean, people believed walking was a good thing, but they didn't know where. Well, it turns out it's different for almost anybody. And you can try to predict it, or you can use feedback to motivate it. So you put a CGM device on somebody, you tell them a 15-minute walk will make a big difference in their glycemic curve, and then you let them discover when to take it. And it's always within 5 to 30 minutes after they eat. When they do it, it's almost as powerful as a drug. And it's consistent and it's easy, and it's something which is safe to do in a very early stage of the disease. The little things like that make a difference, but they're driven by the work that gets you to the real therapeutics too. And which is why I think that work is so important. The work that's coming out of the immune profiling, I think is as important in the preventive space as it is in the therapeutic space. The work Avi is doing is all about getting to people before they have cancer.